All right. Happy Friday. Good to see you guys. All right. Well, any big plans for the weekends? What are we doing? Study. Practice. Yeah. Okay. Anybody going anywhere? Camping? Anywhere? Where are you going, Brooke? Where? Oh, nice. Ministry of Mexico. Very cool. Yeah. Is that just a day trip? Yeah. Cool. Awesome. Very cool. Nice. Good. Well, we actually have a weekend at home, which isn't very often. So we're staying home too, but be good. Well, let's just take a quick look at what we got next week. So everybody's on the same page here. So here's our full schedule, right? We're up here. So we'll have, we'll get into the ear next week. So the way that I always do the exams in this class is the Monday before the Friday exam is the last material for that exam, right? And so then the stuff we cover on Wednesday will be on exam 2, not on exam 1, okay? But exam 1 is next week on Friday. Okay, we will have a reading quiz due or in the first part of class on Monday. I sent you an announcement with kind of details of what pages to read and what figures to know for that. So just make sure you prepare for that. Okay. And then again, if you haven't done the honor lock practice quiz yet, do that. Okay. You might not even want to wait till Wednesday to do it. You might want to do it this weekend because sometimes you'll need to do kind of major things to your computer to get it to work. Okay. And sometimes you're going to need their tech support help. And they're pretty responsive. You use a little chat feature right there in Canvas for honor lock, and they'll help you pretty quickly. But again, you just don't want to be doing that during the exam because then you're wasting your time. Okay. So make sure your computer's ready. That's why I had that honor lock practice quiz due before the exam. But you guys have already been doing it for the most part, right, with the lecture quizzes, so it should be fine. But yeah, so if you've already used it with the lecture quizzes, then you should be good. But still go ahead and do that honor lock practice quiz just to make sure. The other thing that honor lock always has that I always add for the exams is the calculator, built-in calculator. So I don't know if I have any questions where you need that on this first exam, but coming up you will. And so yeah, you just know that's there. But yeah, so that's that for the lab quiz next week. You're pretty much being quizzed on what here. I'll just pull up the lab schedule actually, but you're being quizzed on what you looked at and what you dissected this past week. Here we go. Yeah, so here's the lab schedule. So here we got sheep heart and cat veins, right? So that's going to include pictures of the cats kind of sprawled out. And so those veins up in the thoracic cavity and the arm that you learned, those will all be there. One thing with those is a lot of times it's zoomed in where you can't see the whole cat. So you don't necessarily know, is this a right arm, a left arm? Are we on the anterior side, posterior side? So I'll always put a little image on the slide somewhere that kind of shows the cat's face, and then it has like a little box around where the cat is or where you're at on the cat. So just always look at that first. That gives you your orientation, like, okay, it's the left arm, anterior view. Anterior view is when you see the little whiskers on the image, and then posterior view, you can't see the whiskers. But yeah, so get your orientation first and then try to decide. Don't try to jump into making that decision unless you've actually figured out where you're looking on the cat. Okay. So yeah, so that, and then for the heart stuff, there will be some actual pictures of sheep hearts. Okay. So yeah, so definitely memorize those structures you had to know in the sheep heart. And then there'll be some diagrams too that are a little bit clearer than the sheep hearts for some of the structures that are hard to see on the sheep heart pictures. There'll be just some like actual kind of like textbook diagrams of hearts with some labeling there too. Okay, so those are the big things. And then we're not doing the cat dissection next week. We're doing special senses lab. We will do dissection. We're dissecting the eyeball, so make sure you have your dissecting kit for that. But bring not your anatomy lab handout, but your special senses lab handout, which is the next one down on Canvas. So print those up and have them ready for lab next week. Okay, I think that's it. I do always post study guides for the exams. There's one right above in the module right above the exam on Canvas. It's not super descriptive. It's pretty much just like know everything in the notes, but it also does kind of help you hone in on like, here's kind of the key things that might show up on questions that I call hybrid questions. So yeah, if you didn't have me last semester, you don't know what that means. But pretty much it's a, maybe I'll just show you one. That sounds like a better idea. I'll just show you one real quick. So we're all on the same page. Okay, we're good. All right, so today we're talking about the next senses, which are the chemical senses in the body. So we got the nose doing smell for us, and then the mouth with taste. So we'll talk about each of these. This is a microscopic image of our tongue. Okay, that's what our tongue looks like. All these little spikes sticking up, these little papillae, and yeah, so. And our tongues aren't even that rough compared to like a cat or something. They got really tall papillae sticking up. More cats, yeah, for sure. But yeah, so we'll start out with taste. Okay, so infants have somewhere around 10,000 taste buds. Adults only have around 3,000. And so one of the implications of this is that babies are kind of these little super tasters, and they taste everything really intensely and really strongly. And so that makes me think of my kids. This is Soren, who's now going to be turning 17, and he's my size. But so animals do this too, but babies do this is they feel the world through their mouth, right? They just put everything in their mouth, right? And they everything they can get, they put it in their mouth, and they're trying to just discover the world and learn about it. And so that applies at the beach too, unfortunately. But he would just throw sand in his mouth and just eat so much sand. We were taking the beach, and you try to clean his diaper later, and it was just a mess in there. But yeah, so they're just kind of discovering the world. Part of that is they're training their immune system too, and to get the immune system, that'll make more sense. But just introducing so many pathogens into their body that their immune system has to adapt to all of those and create antibodies against all of those. So they're training their immune system. The other thing that is real common with babies is when you first give them a new food, they make a face like that. It's just like, what the heck? And so it's funny though, like every new solid food that we would introduce to our kids, they just make this face like you just gave them some kind of like poison, like, oh my gosh. And then they want more of it right away. They're like, oh, give me some more. But they have incredibly strong taste buds. And so they're just really tasting everything a lot. But yeah, some funny little pictures there. I just want to look at them for another sec, but okay. All right. Okay. All right. So with our taste buds, we have kind of three types of specialized cells in our epithelium. And so that we have an epithelial layer in our tongue. And so one of those types of cells is just the taste cells themselves. So these are called gustatory cells. And these are the taste receptors. Okay, these aren't the neuron, but the neuron is connected to these. Okay. And so most of these have microvilli that stick off of them. And then on the microvilli is where we have little pores and little receptors. And so the taste pores are little hairs that extend through these pores, and the chemicals pass through these holes, and then they bind to receptors inside there to activate these taste buds. Okay. There's a lot of turnover in these because we're constantly smashing our tongue around, putting in chemicals and hot things that destroy these cells. And so we constantly have to replace these. The second type of cell is just supporting cells. So kind of just cells that are around these cells will hold them in place, give our taste epithelium, gustatory epithelium some structure. And then basal cells. Basal cells are always in the in any epithelium that we have. And these are the stem cells that are there to do the replacing of cells that get damaged. Okay, so those are our three kind of types of cells in this specialized gustatory epithelium. And then the papillae, as are on the back of these slides, are those bumps on the surface of our tongue. And so this is what gives our tongue that rough texture. Some of them, most of them, contain taste buds, but not all of them. But then we also have some taste buds that are outside of these papillae as well. Like in the back of our throat and the roof of our mouth, we have some taste buds as well. And so with the papillae, we have three primary types of papillae. Okay, the first one here are called the filiform papillae. These are the smallest ones. They do not have taste buds in them. These are more just to provide friction, allow you to kind of be able to grab onto things, keep things in the right place when you're chewing and moving things around your mouth. And those are the filiform papillae, they have touch receptors, so we can feel where the food is and move it around, but no taste buds. And then we have the valate or the circumvallate ones. These are big papillae. There's only 7 to 12 of these, but they're kind of in this big V shape on the back of our tongue. And this is where half of our taste buds are found within these papillae. And then we got the fungiform ones and the foliate ones, and these are all just different smaller ones that also contain taste buds, so about a quarter of the taste buds in each of the other ones. Okay, so the main ones that don't have taste buds are the filiform, the rest all do have taste buds. Okay. So let's take a sec and just kind of label these things that we've just been talking about. Okay, so we got different types of papillae, different structures here on the tongue. So go ahead and fill that in, chat with your teammates about it, and you can send in the type of papillae that has the most taste buds out of those options there. Yeah, let me get you another here.
This one's just blue.
I know, I saw it. Yeah. Looks like we got it worked out. The EMT system. Good.
Yeah,
Good.
so hopefully, because I VA counselor insured that even like volunteers for
Later on.
too.
Okay.
Like, or any kind of really a problem.
Good.
So I'm hoping that.
Yeah.
And it comes. We'll see how it goes.
Yeah, good. All right, good for this semester at least. Yeah. Glad you get to stay in there. It's a great class. Yeah.
Oh. Because the size of the tongue one is the foliate. Those large.
All right, so go ahead and send your answer in there on the clickers. Type of papillae that contain the most taste buds. All right, anybody else? There we go. I think that's probably everybody. Yeah, so pretty much everybody's saying A. Okay. So A would be those circumvallate papilla. So they make this little V shape kind of on the back of the tongue. B is going to be the fungiform ones. C, filiform. And then D would be the foliate papillae, kind of on the sides in those folds there. So, okay. And this just shows you those different types of cells that we talked about, right? So we've got the basal cells at the bottom, the gustatory cells themselves with the little hairs, and then we've got the cells surrounding them, kind of supporting the taste buds. All right. Questions on that at all? Okay, well, let's talk a little bit about how it works then. Yeah, so taste buds are kind of in a gourd shape is how they're described. And so that's a gourd. That's what it looks like, kind of big on the bottom, smaller on the top. All right, so then there's a pore on the top to let those chemicals in. And we have these primarily on the tongue, like we were just talking about, but we have some on our soft palate, so like on the roof of our mouth towards the back, some on the inner part of our cheek, some in our throat and our pharynx, and then some on the flap of skin that covers our larynx too, the epiglottis. And so, yeah, we have those gustatory hairs that kind of stick up into the pore, and that's all covered in saliva. So the chemicals that go in there get into solution in the saliva, get kind of broken down, and they can bind to the receptors and stimulate a signal to go back to our brain. Okay, and the next slide has kind of a little clicker type question on it here. So go ahead and take a look at that. And yeah, and so we have the different cell types in the taste buds here. And so you can go ahead and fill that in and send in all three answers just in the right order there, and give you the option to send in more than one. So yeah, talk about that with your team for a sec. Which cells are which? What do they do? And send in those in the right order.
And
Do you call them gustatory cells or receptor cells?
Yeah, either one.
Okay. And then that's a, can't really tell the difference between. Is
Yeah,
there,
that's a good point, yeah.
how does
Yeah. Yeah, I mean, one's probably the hair, or the other one's probably
The
like the
pore.
microvilli, with the other one's the pore, probably. So,
Okay.
yeah, he might be able to kind of figure out which one's the hole.
I'm thinking.
The lower one.
Oh.
I was thinking the other
Oh.
one. I don't know.
Yes.
All right, so send in those answers. Just put the three letters there in the right order and hit the send button on your clickers.
Wait, did you guys? I'm missing D and B. I'm missing A, B, C, D. You just put in order.
So put those three letters in the right order. First one, second one, third one, and hit send. Make sure to hit the send button. All right, I'll leave it open for a couple more minutes for those of you that are still sending it, but let's go ahead and talk through this here. So the ones that are there to support would be D, right? The ones that's the cells that are there to sense the chemicals would be B. Okay, those kind of orange ones. We know that for two reasons. One, because they have a neuron attached to them. Two, because they have the microvilli sticking out. Okay, and then last one, the cells that are there to replace damaged receptor cells would be, in this case, that little kind of smashed blue one there.
So the yellow part is the microvilli, you're saying?
Right here.
Yes.
This is a neuron. So yeah, I think oh yeah, I don't have them on cover actually. So yeah, A is, yeah, we know what A, B, and D are. C is the microvilli.
Oh, okay.
So C is the microvilli. E is just the pore. There's not really a great way to tell the difference on this image between those two, but as long as you know the hairs versus the hole, then you're good. And then F is the neuron, right? So this is a sensory neuron.
So mine do A, B, too, just so.
Say it again. Oh,
Like, you just go
like,
over
yeah,
this.
sure. Yeah, yeah. Yeah, so D, the cells that support and protect. So that's these ones right here. So the support cells. All right, is that what we call them here? Yeah, supporting cells, yeah. And then B is the ones that are actually the sensory cells that had a few names, the gustatory cells or the taste cells. And here I call them the receptor cells, so that the ones that are actually receiving the chemicals, right? So that one is B. So receptor. And then A is the basal cells. Yeah, cool. Questions on that? All right, good. Looks like everybody got in there too. Okay, well, let's keep going. So there's 5 basic tastes that we can taste with our tongue and with the taste buds around. So those are sweet, salty, and sour. Okay, so sweet's going to be sugars, alcohols, certain amino acids, and lead. Salty is ions, metals, sodium chloride being the primary one that we have in our foods. Sour things are due to acids, hydrogen ions, taste sour to us. Okay. Bitter substances. There's a lot of bitter substances. And our mouth is super, super sensitive to bitter substances because most natural poisonous chemicals in nature are highly alkaline. They have a high pH. And so our body's designed to protect us against those. So we taste bitter things really strongly because they could be potentially dangerous. And so lots of things that we might be aware of. Narangan is the chemical that's in grapefruit, and so super, super bitter. And then the last taste is umami, which is like our savory taste. It's kind of a newer one added to this list. And this is most amino acids, a lot of amino acids give us this savory flavor. And so we taste this with meats usually. Keep you guys on your toes back there in the back. Sorry about that. Okay, good. Give me another sec. Okay. So yeah, so it's yeah, there's some other tastes that we might be familiar with, like minty flavors, kind of like menthol or like peppermint, like gum, things like that. And then spicy things too. So minty flavors actually activate cold receptors in our mouth. It's not really a taste receptor, it's a temperature receptor. And then spicy things. They actually lower the threshold for stimulation of our warm receptors, so our mouth feels really hot, and this can kind of spread to our whole body, make us sweat. It kind of tricks our brain into thinking that our body temperature's gotten too high. And so capsaicin is the main thing that does that, that we find in chili peppers. Capsaicin's also in a lot of like supplements and a lot of like icy hot and stuff like that. That's what makes that hot feeling. So it kind of just tricks you. You're not really heating up or cooling down. And so then also the tongue map, which maybe you've seen, used to be what we thought how it worked, that kind of there was regions for different types of taste, but it's not the case. We can taste these different flavors all over the tongue and different parts where anywhere we have taste receptors. So, okay. As we kind of already talked about with kids, it's true also with certain adults are super tasters. And it turns out they have more of those fungiform papillae, and they just taste things more in a more sensitive way, and they might have more strong reactions to those tastes. Again, this narangan was found in grapefruits. Some people don't taste it at all because of a genetic difference. So they can eat the membrane of grapefruits and don't taste anything, which is crazy because it's so strong for most people. Is anybody like that? Yeah, you don't taste it, Ashley? Yeah, not much. Yeah. Yeah, you can just eat grapefruit like, and it doesn't not bitter at all, huh? Yeah, okay. Is that, do you have family members that taste it that way too? You should ask. Yeah, you should ask. It might be a genetic thing in your family. Yeah, yeah. Yeah, and as we kind of already alluded to with kids versus adults, our taste buds decline with age, so we start losing taste and we don't taste things as strongly as we used to. Yeah, Matthew?
Is
Yeah.
that also something that train? Because I know with chefs, there's certain chefs who are very like, they can tell the difference between a lot of different things.
Yeah, there's definitely training, but it's not really affecting your tongue itself. It's affecting the way that your brain interprets things, right? So it's more of like an education of like kind of distinguishing certain flavors that you might just kind of all clump together normally. So it's more of like a learning that happens in your brain versus versus an actual like change in your tongue or your nose. Yeah. Yeah. Ava? Yeah. Uh-huh. Yeah, we'll do that. I think yeah. Yeah, I think we'll still do that lab. We used to do a lab, and I think we still do it this spring. You'll do something like that in the lab. Yeah, yeah. Or at least we used to. I can't remember if we removed that one, but yeah, totally. Yeah. Okay, so then the taste pathway, once we activate those those cells in our tongue, then that causes depolarization. Okay, some of the tastes use the what's called the second messenger system. Right. So the second messenger system is we bind something to the receptor of a cell, then it sets off this chain of events, usually involving a G protein. We talked about this a little bit last semester. And then all these things happen, then eventually we get an action potential on that cell. So that's the second messenger system. But then some things can just rush right into our cells, and we can get an action potential. Okay, so we'll talk about that a little bit more in a sec. But overall, we end up getting an action potential on the sensory neuron somehow by that receptor cell getting activated. And then it goes up to what's called the gustatory cortex, which is in the frontal lobe of our brain. Okay. And so we kind of have, we have a few different, three different cranial nerves that carry that signal up to our brain. Okay. So on the anterior 2/3 of our tongue, it's our facial nerve, cranial nerve 7. On the posterior 1/3, so the back of our tongue, it's glossopharyngeal, that's cranial nerve 9. And then the other ones around, like our mouth and our throat, are carried by the vagus nerve, cranial nerve 10. So it goes from our tongue through the medulla and the thalamus and then up to the gustatory cortex to be interpreted in our frontal lobe. Yeah, once you're done writing that down, feel free to flip to the next page and start thinking about that question and talking about it with your teammates. We'll come back together in a sec. All right, let's come back together. So who remembers how action potentials happen? Raise your hand if you think you remember how action potentials happen. I know some of you guys do. What's the thing that causes depolarization? Sodium doing what? Going in, right? Going into the cell. So the body takes advantage of that mechanism. And when we eat salt, we dissolve sodium chloride and sodium rushes into the cells and we get an action potential right there, right? So it's kind of the same exact mechanism, but that's what makes us taste salty food, right? We see that same thing with some other chemicals as well, like potassium, like hydrogen ions. For the hydrogen ions, that would be the sour foods, right? And that just allows hydrogen to just rush right in, depolarize the membrane of that cell, and then activate the sensory neuron, right? It's all about just starting a signal on that sensory neuron. And so different ways that the body takes advantage of doing that. So that's kind of the direct mechanism, right? And then there's other tastes like umami, where the amino acids bind to a receptor on the cell, set off a chain of events that eventually leads to the activation of that neuron. But it's not direct like these ones, kind of. So two different options when it comes to taste. Yeah, Ethan. No, just the idea of like sodium rushing in, hydrogen rushing in. That's fine. Yeah. Okay. All right. So then let's switch gears over to smell. So with smell, we have another epithelium, and this is called the olfactory epithelium. Okay. So specialized epithelial cells in those concha of our nose. Okay. Not too much area, about 5 square centimeters of area. But we have about 10 to 20 million of these receptor cells. Okay. Way more than we do of taste cells. Okay. The one interesting part about smell is we really do have to kind of sniff forcefully to get the air to hit that part of our nose where the receptors are, right? And we all do that, right? When we try to smell something, we go like we kind of do that, right? And that's because it creates some turbulence in your nose and makes the air, instead of the air just doing this, right? That misses the receptors. We have to kind of make it go up that way, right? To where the receptors are. Dogs do this all the time, right? Eventually, I'll bring Betsy in for a test, and she'll sniff all your feet. You'll hear her all the time. She'll be grunting all over the place. But right, you have to actually do that. I bring my dog in for exams. For those of you that didn't have me last semester, but she's the stress relief dog. But yeah, but we got to do that little sniff to make it hit the receptors. So yeah, so the types of cells very similar to what we talked about before. So we got olfactory receptor cells. These are bipolar neurons that are constantly turned over. And then we've got hairs as well, olfactory hairs, cilia on the end of those. That's what's the chemicals are gonna bind to. Supporting cells, just like we saw in the mouth, right? So these are surrounding those olfactory cells, holding them into place. And then the basal cells, just like in the mouth as well, right? Cells that are there to replace the damaged receptor cells. So if you're done filling that in, you can just flip over to the next page and label that diagram. There's a clicker question there as well. All right. Yeah, go ahead and work with your team labeling this, and there's a clicker question there for you to send in. You guys are living the life that these students want to live someday. They're looking at you like, I want to be that beer.
They don't know we're doing an hour.
It's true. Yeah. It's true. Yeah. Please.
Okay, what do you mean by hairpin turn?
So you know what a hairpin is? Like a bobby pin, like a U-turn pretty much. Yeah, so it's the same idea. Like if the air just can kind of go this way down your throat, it has to go like up,
Okay,
almost like make a U-turn to get to where the receptors are.
all the receptors are in.
They're in like the top
superior
part.
check.
Yeah.
Okay,
Yeah.
thank you.
Yeah. Do you wear bobby pins, Ellen?
I literally have.
You going in right now? Yeah. Didn't know if it's still that much of a thing with girls, but yeah.
Not just like how are we able to like train and like just
Yeah, so you mean, well, specifically for taste, and from our tongue, so it goes to a different part, each of those. So like the neuron that's attached to a receptor that's gonna receive sodium. So there's certain ones that receive salt, certain ones that receive, like each of the five flavors, there's different types of receptors for each. And so those are going to activate different neurons, right? And so then there's a group of neurons that are all going to go the same place for salty flavor in the brain, right? And then there's another group that are going to go to sour flavor, right? And so it's kind of like same thing with our eyes. Like when a certain color is activated, it goes to a part of our brain that the brain knows it's red or whatever, right? And so, yeah, it's just like that perception piece. But then when it comes to taste, we really use our nose almost more than we do our mouth for a lot of flavors. And so that's part of it too. And then it's just really like training your neurons to associate certain chemicals with a flavor, you know?
It's like and dislike.
Yeah, that's a good question. Yeah, yeah, totally. A lot of it's definitely like emotional reactions and associations with food. Like if we had a bad experience when we ate a certain thing, then we might never eat it again, right? And that's part of like the learning that happens in your brain with foods. And then yeah, so there's some of that. There's some just cultural things like where we've been trained to eat these foods our whole lives, and so it tastes good. Like we were just in Taiwan and they ate stinky tofu. Have you ever smelled that stuff? It's like smells so bad. And they're just sitting there eating it. I'm like, how are you guys eating that? But they've been around it their whole lives. So it's like tastes good to them somehow. I'm just like, yeah. So there's a lot of factors. But yeah, good questions. Yeah. All right, send in your answer here. Go ahead and hit the send button. Since there's more than E as an option, got to use the send button for this type.
Yeah. So we're just using B. Wait, it's good. Back to the presentation. Yeah, that's low key in example, back to the track for that one, but it's already legal.
All right. I'll leave it open for another minute here, but go ahead and just hit send just in case, because there's about 4 of you missing still, but we can start walking through this. So most of you are saying F, and then a group of you are saying B as well, but let's walk through it. So A is what's called the olfactory bulb, and you guys might remember this from the sheep brain that you dissected last semester, right? Where we got to see those olfactory bulbs. Okay, so it's just kind of like the end of the olfactory cranial nerves, right? Kind of group up into these bulbs, and then from there, they're going to go through these little holes. You guys remember what bone this is? There we go. Maybe. Yeah, the ethmoid bone. Yeah, we got the ethmoid bone and the cribriform plate. Remember, it had all those little holes in it, right? The holes are these holes, right? So the neurons can get through and connect there. And so, yeah, so that's that. So we got the axons of those olfactory neurons sticking through those holes. C would be the olfactory neurons themselves. Those are the bipolar sensory cells. Bipolar because they're kind of, the axons are kind of the same on both sides, right? That's why they're called bipolar cells. And then D would be those supporting cells holding everything together. E would be our basal cell again. And then F is going to be the olfactory cilia, right? And that's really where the receptors are, is on the cilia. And so that would be the correct answer for the question here. So this is all a mucus in here, right? And so the mucus dissolves the chemicals, and then those chemicals can then bind to the receptors on those olfactory cilia. Okay, questions on that? You got it? I'll give you a second. Yeah, go ahead. All those things, yeah. Okay, good. All right, so smells, we can detect somewhere around 10,000 different smells. Okay, so way more complex than our taste in our mouth, right? And so neurons can be stimulated by different combinations of a lower number of chemicals, right? So maybe it's somewhere, they don't really know, but somewhere between 7 and 50 different distinct chemicals, but it's the combination of all the different possible combinations of those that give us the 10,000 different smells that we can interpret. And it's much harder to research. Number one, because it requires a substance in its gaseous state, which is always more dangerous, right? And it's always potentially it's always hotter for one thing, right? And it's potentially can get to places that people can inhale it and it can hurt them. And it's just harder to hold on to and grasp those chemicals. And so it's just a little bit of an area that's less researched than taste. The pathway here is that molecule is going to bind to a receptor on one of those cilia. It's always going to kick off the second messenger system. So we don't have any options where it directly stimulates it like we did with taste. Okay. That's going to open an ion channel. We're going to depolarize that neuron, send our signal from there. That olfactory neuron is going to stimulate one of the neurons from the olfactory nerve, cranial nerve 1, right? In that bulb. One of the tracks in our smell sensation goes to the temporal lobe, and then the other track kind of goes through all kinds of places. It goes through the hypothalamus, so we can kind of have emotional connections to smells. Goes through the limbic system, which is connecting smells to memories and emotions, as well as the hippocampus, which is important for memory as well. So we all know as humans that certain smells can take us right back to places, right? We can smell something and it's like, whoa, I feel like I'm literally like in the hospital in this moment that I had with my family or whatever it is, right? And it's because our smell is so intertwined with the memory centers and emotional centers in our brain. So yeah. So quick question on that. It's about some research that was done in this area. Go ahead and read through this and chat about it with your team for a sec and send in your answer.
That was just thinking one before. Yeah. I don't know. What was it?
Yeah, there's crazy stuff. It's not even really. Oh, yeah, well, too far. We don't think it's right?
No, there's some evidence of it still in humans. Yeah.
Really?
Yeah, for sure. Yeah.
Dang.
Yeah, there's
'Cause I was
certain.
always curious of I was looking it up, but I didn't get anything crazy. I was thinking,
Yeah.
you're just like,
It's not like super, yeah, it's not super strong like it is in other animals, but there's definitely evidence of even women getting on the same like stage of their cycle together when they're living together and some pheromone related. And
Dang.
yeah. So it's kind of weird. Yeah. All right. Send in your answer if you haven't yet. All right. About 5 more of you need to send it in, but about 2/3 of you are saying A and about the other third saying B. So when you think about a right-brained person, what does that typically mean? They're what? They're cooler, yeah. They're cooler. Yeah, right-brained people are typically like artsy, maybe a little bit more emotional, maybe whereas like left-brained people are the kind of sciencey and very exact and specific, right? And that's not just like a thing that people talk about. There is science to that. And so the right brain has more emotional connections to it. And so remember with our cranial nerves, it doesn't cross sides, right? Like with our motor neurons that contract our muscle, we're going to use the left side of our brain to contract the right side of our body. But with cranial nerves, we go directly to it. So there's no crossing, right? So the right nostril goes to the right side of the brain, left nostril goes to the left side of the brain. And right's going to have more emotional connections. Yeah. And so then it wasn't just this, but then they were more accurate with the left side, actually, in identifying the smell. They were more accurate with the left nostril. So kind of interesting. Okay. Questions on that? Okay, so we have some individual differences with smell as well. So some genetic differences. Some people can't smell cilantro at all. And if you can't smell cilantro, then it tends to just taste really bad like soap. Yeah. Yeah, so which the more extreme reactions would be the right brain, but then the more accurate in interpreting what it is would be the left brain. Yeah, yeah. So right brain people are more artsy and emotional, whereas left brain people are a little bit more scientific and exact and specific in general. Yeah, yeah, yeah. So does anybody think cilantro tastes like soap? Anybody have that experience? Yeah, yeah, it's a genetic thing where you can't actually smell it, so you don't get the smell of cilantro, which is the best part. Otherwise, it kind of just has a weird soapy texture. And yeah, and so then there's this other chemical, ethyl mercaptan, which it has this garlicky, rotten egg smell. And it's actually put into propane. That's what's in propane. Propane has that rotten egg kind of garlic smell. It's a chemical that's put in there just because natural gas doesn't have a smell normally, and so it's dangerous that it could be leaking and people wouldn't know it was leaking. So they put that stuff in it, but certain people can't smell that stuff, and so there's a problem there. Yeah, and then as similar to taste, your smell declines with age too. So did anybody get the symptom of COVID where you couldn't smell, couldn't taste? Yeah, quite a few. Did anybody still have like did it continue? Is it always temporary? Yeah. I've had some students have known that like lost their sense of smell with COVID and never got it back. And that definitely happens sometimes, but yeah, rough. So most of eating, even though we have the taste in our mouth, most of our taste is actually through our smell, right? So yeah. Okay, so this idea of anosmia, oops, anosmia is when we either have a reduced or a loss of smell completely. This can be a congenital thing where our olfactory bulbs don't develop properly, or it can be caused by trauma. So this could be facial fractures where we destroy the cribriform plate and it slices all those neurons. So that could be car accidents, other things like that, where somebody's face just gets smashed, right? And then certain chemicals that get in and actually destroy those basal cells. If we destroy the stem cells that are there to replace the olfactory cells, then we can't replace them and they don't really come back. And that was part of what COVID did, was able to get into some of those cells and damage the receptor cells, but and then in extreme cases destroy the basal cells as well, so they could never be replaced. And there's a couple other things there, a couple other degenerative diseases in the brain that can cause misinterpretation of smells, as well as just loss of smells. And then zinc deficiency is a big one too. Okay, one more clicker question. Go ahead, chat about that with your team and send in your answer when you're ready. Give you a sec to finish writing this. All right, check your answers with your teammates as well. Make sure you're on the same page. Send it in when you're ready.
Maybe your teeth. Yeah, this was called. Those directors.
All right, go ahead and plug those answers in, hit the send button just to make sure. All right, I'll leave it open for a sec. A few more of you, I think, just need to hit send, maybe 3 or 4 more. Go ahead and do it just in case. So yeah, most of you are saying C, D, and E. So taste, it can go either way. We can have direct stimulation or the second messenger system. Smell is always the second messenger system. So that's correct. All right, guys. Have a fantastic weekend. We'll see you guys on Monday.
Is this on, it's and this one, right?
Say it again.
The,
Oh, yeah. I think so, yeah. Wednesday and Friday, I think. Yeah, you can just look at it. You can click on it and see. Pretty sure. Yeah, because the all day, right? Or no, Wednesday was PBO. It wouldn't be on that one. So it was on last week. Yeah. Yeah. That's what I mean. Yeah. Yeah.
So for the final,
Yeah.
is it cumulative since last semester? Oh, I just scared my. They were like,
Chemistry's that way, yeah. Yeah, that'd be mean for this class. Yeah. All right, have a good weekend.
No, me too. I had to ask one.
Y'all have a good one. Have a good weekend. See you Monday.
Yeah.
Already took care of that, request. Thank you for.
Oh,
sending it
good,
to
good,
me.
and then good. And then, did you get into the class too?
I don't know.